Today we're talking about lipids, the fabric of life. And so you see a picture of a yeast cell on the left side. And so that's the image that you can think of as life. We study life at a scale below that can be seen by a microscope. And so we are left with cartoons, our imagination, hopefully an imagination that's based in data. So we're studying the tree of life. So we're studying life in general. And so I think about life, think about evolution. And we go about 3+ billion years of history to understand. And that's a lot. So we're looking at the very tips of essentially 3 billion years of events, which involve organisms reproducing with genetic variation, offspring generated, those offspring surviving or not surviving, reproducing with genetic variation, and so on. So we're dealing with these honed mechanisms to deal with all sorts of environmental challenges. So we use yeast as a model system. It's a eukaryote, it's a single-celled organism, but the idea is that a lot of events that are central to cell function developed very early in evolution, so there'll be precedents set in this organism that will be still found in humans and frogs and butterflies. So very conveniently, after the last talk, we're talking about barriers. And cell membranes provide a barrier to allow for a very controlled situation where cells can import specific molecules and retain specific molecules. And this is central to cell function. It also allows for cells to have compartments. So within cells, much like rooms in a house, there are different environments where different events can occur. And there's, again, transporters embedded in those membranes to allow for careful transport in and out. Should also mention that cell membranes have proteins embedded in the cell surface, which allows for molecules also come in and out of the cell. And so there's receptors, there's transporters, all these dynamics and important functions that occur at the and within these membranes. Also important in cell function is this process called endocytosis, where membranes can eventually invaginate, form even more invaginations, eventually this neck combines to pinch off. So it's a lot of structural requirements for these membranes for appropriate cell function. And these membranes are not like fabric, we think of, say, cotton or wool, where there's horizontal strands, but they're actually largely very small vertical pieces that can flex. And we'll talk about that in terms of the phospholipids, which is the molecule I want to talk about. But still, I want people to have a big picture of what we're talking about before we jump into the biochemistry, about cells, about this barrier between the inside and the outside, about barriers inside. In this case, the barriers are good, allows for control, because cells are controlled places. Otherwise, life doesn't go so well. So the best analogy I come up with for the fabric of life of membranes is chainmail, which has its issues as well. So it's not a great analogy, but it's flexible. It's made of small parts. So perhaps you start getting to the ballpark of what I want to talk about. Now we're getting into the extent of how people can view things. And so there is something called atomic force microscopy. Again, this is other people's work, where essentially you look down on the surface of a membrane from above, much like a plane looking down on the ground below. And there's this methodology used to essentially determine the topology of that surface. This is an artificial membrane where a variety of lipids were mixed together and then viewed. And it turns out these lipids will self-associate, not just to form this bilayer, which I'll talk about soon, but also different lipids will cluster with each other to form different subdomains or microdomains. So it looks like it's like a Rorschach blot, but essentially the lighter domains are higher, it's like trees off the ground. The darker domains are lower, so like grass closer to the ground. And so it's just an idea that these lipids do not form homogeneous mixtures, but it's distinctly heterogeneous mixtures with subpopulations. Again, that's consistent with what people have seen actually inside cells. This is an artificial membrane. But now we're actually beyond what we can see with a microscope or any visualization technique. So we're left with cartoons. Again, we do biochemical experiments, and we try to visualize them and relay those through these cartoons. And so phospholipids, shown here as almost a balloon with a head, which is orange, and these two tails in black. We'll see the actual structure of them soon, can self-associate based on their structure in this way. But they can self-associate in a variety of ways. If there's a lot of them that they're closely packed, they can form what's called a gel. If they're loosely formed, they form a liquid ordered. If they are more loosely formed, it's liquid disordered. So there's a variety of these forms within any given cell membrane. It's not that all in one cell membrane. So getting away from cartoons, here's the actual organic structure, organic chemical structure. And this is really central to what we're trying to do here. And it's not so easy to interpret. These squiggly lines are hydrocarbons. So one carbon linked to another with hydrogens coming off the side, very hydrophobic. So there's two of those. Each phospholipid species shown here has two of these acyl chains, which is consistent. There's a chassis essentially, or a 3-carbon backbone onto which all these molecules are bound. So for each phospholipid, there's a chassis, the glycerol backbone, 2 of the acyl chains, which are hydrocarbons, and a third structure called a head group. Head groups are small, and they can change depending on their charge of positive or negative and their size. And so this creates a lot of possible heterogeneity amongst phospholipid species because the acyl chains can vary in length, typically 14, 16, 18, or 20 carbons long. They can vary in the number of double bonds they have. Here, each one is shown to have 1 double bond. They can be up to 5 in a single acyl chain. Each double bond changes the orientation of that straight chain to sort of a kink. And so it creates a broader structure to that phospholipid. So while there's a fairly simple structure, through combinatorial diversity, there can be hundreds of different phospholipid species found in any given membrane. Mammalian systems tend about over 300 to 400. Simpler systems like yeast, more like a little over 100. In the future, I try to get away from the balloon and 2 strings dynamic to the stick diagram shown below, where you got the chassis, the vertical part, which is that glycerol backbone, the acyl chains coming off to the right, and the P being a phosphoryl group, and the head group being this small group, could be positively charged, could be negative, could be both. Yeah. So this nifty technique called electrospray ionization tandem mass spectrometry. It takes longer to explain than to say. It's a analytical chemistry technique to take a lipid population and to categorize the abundance of all these lipid species, because they each give a unique peak, and we can measure the area under the peak. In fact, we don't do it. We collaborate through a wonderful group in Kansas for a fee. And so we give them the money, they'll analyze all the samples we want or that we can pay for. And so essentially this technique allows you to take, say, a lipid sample and characterize what are the relative abundances of all these different species. In yeast, there's about 100 different species. Just shown here, I picked 27, the most abundant ones, to show that they vary based on the head group, based on the 2 acyl chains together. Because remember, their lengths are about 14, 16, 18. So by lengths together, you get 28, 30, 32. And so that's essentially what's trying to be shown here with the bar graphs indicating the extent of the abundance. If I'm running out of time, okay, so I need to go faster. These are made by a synthetic pathway. And there's 2 steps. One's called GPAT, one's called LPAT. One adds the first acyl chain, one adds the second. The other enzymes I'm not going to worry about. So we want to study them because in yeast anyway, there's 2 enzymes that meet the first reaction, 2 enzymes do the second one. In humans, there's like 4 for the first, 5 for the second. So it's more complex. We're using a simpler system to understand a more complex one in humans. We can use yeast to make knockouts. We can delete different genes and then look at the effects. And the effects here are the phospholipid compositions when we knock out these various genes, which again is multifold. You see a lot of the bar graphs. How do you analyze all this? Very hard to do. So I collaborated. Collaboration is great. Kishal Pokral is an assistant professor in the Department of Mathematics and Statistics and helped me greatly. In fact, he did all the work to analyze this data. And we came up with this idea of heat diagrams to show the relative abundance of the lipid species. So we got about 110 different phospholipid species to compare the abundance of in 5 different scenarios. 1's wild type and 4 of the double knockouts. And so it's a lot of data to go through, but it's a whole lot of work. And so this is the raw data, and we're going to jump through that and just focus on the very top part of the middle graph, and that is, if you look at the phospholipids based on acyl chain length, there's a distinct difference amongst the different yeast strains, suggesting that the different enzymes have a different impact on how long the acyl chains are. And I think I'm running out of time here, so I'm trying to be kind of quick and perhaps too quick. But we've identified this interesting dynamic where different enzymes contribute to different lengths. And the idea is to say, well, will this also be true in humans? And the nifty thing is that the one enzyme that came out of all this is SLC1, has a nifty thing of substrate specificity. If I'm over, just turn me off and I'll deal with it. But essentially, since the enzyme has two substrates, one is the lyso-PA, again shown here with the stick diagram. One is an acyl-CoA, right? Both have different chain lengths. So it's trying to like fit two cars into a garage. And the substrate, the enzyme binding site is not quite like a garage. It's not quite so square and rectangular. It's all irregular. It's got charge distributions. So different molecules can fit in to different extents. So if the enzyme has certain specificity, can fit, say, the one substrate first, and one car is big and one car is smaller. And it seems like the garage can only fit one big car and one small car. So it's not that it only fits big cars or small cars, it can fit one of each. And so if the first molecule, the one with has this shaped like a right angle, has a long acyl chain, it'll use a short one next. Alternatively, if the first one has a shorter car, the next one will be longer, which is unique. No one's ever shown that before, and with maybe a real neat way for one enzyme to have a distinct impact on the phospholipid composition of cells. So we're hoping to extend this to humans, and we thought that'd be neat. And so we pitched this idea to the NIH. We wrote a grant they have a lot of institutes there, but fortunately one of them focuses on basic science, because that's what we do. We're not studying necessarily viruses or cancer, we're understanding how cells work. So one of those institutes is the Institute of General Medical Sciences. And they have a special grant program for universities that give out very few PhDs and don't have a lot of grant money, and that's us. So it's our R15 program. That's the title of the grant. I was very fortunate last year to get the grant. So we got all this money to pay all these undergrads to do research and to pursue these projects. Now with COVID, we're kind of slowed down with that, but we're hoping once things get really cranking again, being optimistic, that soon we can really got a lot of experiments lined up to do. So with that said, I want to thank all the undergraduates who worked with me in the past and been central in getting up data. So convince NIH we can do this in an undergraduate environment, do some real science. And certainly thank Kesha for a wonderful collaboration we've developed. We've got 2 papers together now, and Ruth Welty at Kansas. So I'll stop there. I talked way too fast. I hope you learned something. I'm happy to answer any questions.